One of the main things here is that one of the issues here, or one of the heights of this part, is that let me get the pen working. Okay, the epidermis, like I told you, is transparent, right?
Yes.
What did I say about it?
Since it's transparent, it allows for light to pass through
Thank you.
easily.
It's also protective. It protects the mesophyll cells. And again, another thing about it is that it does not, the epidermal cells does not contain chlorophyll. No what?
Okay, no chlorophyll.
No chlorophyll or chloroplast. What does that tell you?
The photosynthesis doesn't take place.
Thank you. Photosynthesis doesn't take place there. But the guard cells have chlorophyll in them, or chloroplast, a few chloroplasts, okay? The guard cells have chloroplast in them, meaning some photosynthesis can take place there. All right. So take note of that. These are some extra details that might trick you a bit when you see questions on them. We've also tried to describe the guard cells. Oh, you can even see the chloroplast there. Okay, of the guard cells. So we've also talked about testing for photosynthesis. Let's talk about varying light intensity. Varying light intensity. Look at this setup. Um, just quickly look at the setup and try to tell me what you think is happening here. It says everything here is talking about light intensity and the rate of photosynthesis. Just look at what is on the screen and tell me what we can deduce from it. Let's see. There is the
Um,
ruler.
they are putting the lamp closer to
Mm-hmm.
the plants, so
Mm-hmm.
the light intensity can increase and increase the chances of photosynthesis in the plants.
Yes, so the closer the lamp is to the light, of course, the higher the light what intensity. Okay, higher intensity will definitely increase the rate. Will definitely increase the rate of photosynthesis. Now, there are different ways to alter the rate of photosynthesis. A lot of factors, or a few factors, can affect the rate of photosynthesis. One main factor,
Oh yes, we learned it in school. I remember learning
yeah,
this about three
okay,
weeks ago. We learned about light intensity, carbon
mm-hmm,
dioxide concentration,
thank
temperature,
you,
and
mm-hmm.
leaf surface area.
Area, beautiful, beautiful. So let's be a bit more direct. Now we know light intensity has an effect on photosynthesis. Temperature is another, okay. Carbon dioxide concentration, okay. The more carbon dioxide available, the higher the chances of photosynthesis occurring. And of course, the wider the surface area, the more light the plants can get, the more light the chlorophyll in the leaf can absorb, and the higher the rate of photosynthesis. Now, the question is, how does temperature really get to affect the rate of photosynthesis? What does temperature actually have effect on that hint on photosynthesis?
Um,
Let
uh,
me help you. I don't know if you've done something on enzymes, right? You've
oh
learned
yes. Oh,
enzymes.
Sasa, you just reminded me of something. So
Yeah. Okay.
at low temperatures, enzymes
Mm-hmm.
activity may be reduced, which can like
Mm-hmm.
slow down photosynthesis. But at high
Beautiful.
temperatures,
Mm-hmm.
enzymes can be denatured, which can speed up the rate of photosynthesis?
Yes, you said that high temperatures, enzymes activity could be what? Enzymes become what?
Denatured. Denatured.
Mm-hmm. So
Which
on denaturation, it means the enzymes have lose their shape. Once they are denatured, they can't catalyze reactions anymore. So what we are saying is when the temperature is very low, the activity of enzymes slows down,
okay.
right? When the
Mm,
temperature is optimum, that's the word. We've heard of optimum temperature, right?
yes.
Did you hear of that term?
Optimum
Do you
temperature.
have, yeah?
No.
Okay, you don't know optimum temperature. Okay. Optimum temperature is more like what we call an ideal temperature. The temperature at which the activity of an enzyme, okay, the
Mhm.
activity, if this is an enzyme action, okay, something is wrong here. Okay. If this is the rate of an enzyme action, so if this is the rate, and that's temperature,
Okay.
at the start, when the temperature is low, the enzyme activity is also low. Okay.
Okay.
But as the temperature increases towards the optimum temperature, the enzyme activity is at the highest, at the peak. So optimum
Mm,
temperature is referring to the temperature at which the activity of the enzyme is at the peak. It's more like the best temperature at which the enzyme functions. So above optimum temperature, further increase in temperature when it becomes too high, above optimum temperature, the enzymes begins to get denatured, right? And the activity drops towards zero. Okay, once it drops finally to zero, it means the enzyme is totally denatured. So extreme temperatures denature enzymes, and when they denature the active site shape of the enzymes, the ability of the enzyme to catalyze photosynthesis reactions also slows down or drops until getting to zero. All right. So what temperature does is that when it's too low, the activity of the rate of photosynthesis slows down. When it's optimum, it's at the highest. Above the optimum temperature, it drops again. So that's how temperature affects enzyme action. Do you get that?
okay. Mm.
Do you understand what I've explained now? Do you understand what I've explained now?
Yes,
Are
sir.
you
Yes. Yes, sir.
okay? So that's how temperature affects enzyme action. For light
Okay.
intensity, that is the main point on hand. The closer the lamp to the plant, the higher the intensity. And if you look at this table of results as seen on my middle right hand corner, the table of results, as the lamp was being brought closer to the plant, at first it was 1 meter away, then 0.5 meters away, then 0.25, 0.125. What can you say is happening to the number of bubbles counted per minute?
It increases.
It increases. And what's the relationship between the number of bubbles being counted per minute and the rate of photosynthesis?
Um, as the bubbles increases,
Hmm?
as the bubbles increase, or
Mm-hmm.
no, as the bubbles increase, the rate
Mm-hmm.
of photosynthesis
Just
is
calm down. Yeah.
is
Also increases. That's
yes, yes.
just it. So the number of bubbles given off shows that there is an increase in the rate of photosynthesis because the bubbles definitely are oxygen gas bubbles. And oxygen gas is produced only when photosynthesis is happening on the leaf. So if we get more bubbles per minute counted, it means the rate of photosynthesis, of course, is getting higher within the minute. So that's just the link between the number of bubbles counted and the rate of photosynthesis. So from this experiment, if I ask to state the conclusion of this experiment, now we can say increase in light intensity definitely increases the rate of what? Photosynthesis. They can ask you what value, what result from the experiment shows that increase in light intensity increases the rate of photosynthesis. All you just say is because of the increase in number of what? Bubbles as the light intensity was being increased. But look closely at the table. Something happened here between 25 centimeter mark and 12.5 centimeter mark. What happened to the number of bubbles? Can you see the table closely?
It stays the same. Okay, so
It
maybe
stays the same.
it's
Can
at
you give a
its
reason for that?
optimum. It's at its optimum. Was it temperature or?
Remember, we are referring to light intensity. So the answer should relate to light intensity,
Oh,
not temperature.
hmm.
Go on. I'm with you.
So I'm thinking.
Go on. Okay, let's say you have 3 boxes of pizza to eat. And because on a normal day, you are not selfish, but to that day, you just decide to be selfish. You decide not to share with anyone. It's of course the very big size. You've eaten the first box. You are down on the second one. And by the time you are getting halfway on the third one, your tummy gets filled such that you can't even swallow anymore. What do you think has happened to you at the time?
You've reached like optimum capacity where your stomach can handle
Can
all of that
contain, right?
food.
So what's
Yes.
happening to this plant here now? The chlorophyll in the leaf, what do you think is happening to them? Because they are the one absorbing the light.
Or maybe the chlorophyll has um there's not like too much chlorophyll inside the leaf. That's the amount. It has reached the amount that it can contain.
Thank you. So it means that the chlorophyll in the leaf has absorbed the light to the maximum they can take within the unit of time. So any further increase in the light intensity will no longer increase the number of what? Bubbles. Even if you increase the distance of the, or if you bring the lamp much closer, let's say to 10 centimeter, okay, a bit more closer, the number of bubbles most probably will still remain at 105 because the amount of chlorophyll in the leaf can only absorb that much light within a minute. And so the number of bubbles will probably stay the same all through. At that point, light intensity is no longer what we call a limiting
Factor.
what? Factor.
Factor.
All right. So limiting factor is any factor which when in short supply can affect a life process. At the start of this experiment, when the light was 100 cm cube away or centimeter away rather, light intensity was the limiting factor. So the closer you bring the light towards the plant, the more the bubbles will record because it was limiting. But from 0.25
Okay.
down towards 0.125 distance closer, is showing that light intensity is no longer a limiting what? Factor. You get the idea
Okay.
about limiting factor and photosynthesis now.
Yes.
That means something else, some other factor is limiting. They could ask you, mention 2 other factors that could be limiting in this experiment as we stand, as it is now. If further increase in light does not increase the rate of photosynthesis, what do you think is limiting in this experiment? What could be? Yeah.
So can you come again, please?
I said, since we have established that at this point, light intensity is no longer a limiting factor for this particular experiment, What other factors could be limiting for this experiment?
Oh, other
Suggest,
factors that for this
give me name.
experiment.
Yes.
Maybe carbon dioxide concentration
Thank you. Maybe carbon dioxide. Perhaps there is no more carbon dioxide available for the
and
plant to
temperature.
use for.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have um, you know the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, um, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable, and you get the very best of service. We also have for the E levels as well, um, and also for the EdExcel. So we have for the Cambridge E levels and EdExcel E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com, and they'll reply to you. And the good news about this is what I've discussed with the sales team is we've tried as much as possible to see how we can subsidize the amount according to countries, so it doesn't really look very, very heavy. Okay, so this is really important. Yes, we do charge in pounds, but again, we try to be as much as possible considerate to your countries, and we don't just skyrocket the price. No, because at the end of the day, we know how it feels, and we really want you to actually get the best of service, and again, be able to afford it from the comfort of your home. So again, Ajiro Tech private tuition is currently ongoing. You can enroll yourself for the IGCSE, and then for the E levels. Again, for the IGCSEs, these are the subjects, and for the E levels, these are also the subjects. So again, what are you waiting for? Just send an email to the system. If you're an IGCSE student preparing for IGCSE, if you're watching this in 2025, watching this in 2026, watching this in 2027, we're currently ongoing, and this is actually a good time for you to actually explore, and then get it tailored to your own needs. So trust me, you don't want to miss out.
What?
And temperature.
Yes, perhaps the temperature is no longer optimal. Okay. And the surface area of the leaf could be a factor. But there's what we
Yeah.
call environmental factor, right?
Okay.
There are environmental factors. There are also internal factors. So you have to be wary or careful of what factor you are doing now. See, they ask you to mention environmental factor, then carbon dioxide concentration should be a good answer. Temperature could be a good answer. Some can talk about amount of water available. Okay, amount of water available. Perhaps there is no more water for the plant to use for photosynthesis. Water is a raw material. Okay. And when we talk about internal factor, then you cannot talk about the surface area of the leaf, right? surface area of the leaf. You talk about the amount of chlorophyll, amount of what? Chlorophyll, okay, and the plant. Those are internal factors. Any question?
No,
Any question? No. So that's
no.
just a brief on light intensity, and I've tried to introduce limiting factors for us. So most times, both for paper six and in theory questions, they can play around these ideas, and you need to think outside the box. Usually, those questions are more of scientific inquiry questions. They are more of scientific inquiry questions. Okay, questions that
Okay.
will test your knowledge of carrying out scientific procedures or processes or activities in probing and finding out answers to hypotheses and stuff. Now, having said this, you could also talk about factors that we need to keep the same, factors that we need to keep constant, factors that uh, that we need to change to make our investigation fair or make our results accurate, or that will not make our results to be affected. All these things, like for this experiment now, what factor do you think we have to keep the same in this experiment? What factors do you think we need to keep the same so that the results of our experiment or this investigation can be fair?
The light intensity.
We're already changing the light intensity.
Oh,
So
oh,
what are the things that we must ensure are to be kept constant for our experiment to be fair, that we
oh.
keep available? Yeah.
Water availability.
One, what else?
Uh, Carbon dioxide concentration.
Thank you. Carbon dioxide concentration. What else? To also maintain the
Temperature.
temperature, because temperature could be a limiting factor. Okay, we have to ensure that there is constant supply of carbon dioxide. We have to ensure that okay, yeah, I think this okay, and amount of water available. One way to ensure that there's constant supply of carbon dioxide is by adding sodium hydrogen carbonate to the water. If you add sodium hydrogen carbonate powder or liquid into the water, it increases the amount of carbon dioxide available in the water. That will make carbon dioxide not a limiting factor. Okay,
Okay.
do you get
Okay.
that now?
Yes.
Yeah. So sodium hydrogen carbonate is what we call, I think, soda lime. Yeah, I think soda lime. Okay, it will increase the amount of carbon dioxide in the water. We can keep the temperature of the room regulated so that the experiment will be fair. All right, we can ensure that there's enough supply of water, okay, for the experiment. So these are the factors we need to keep the same for the reaction or the experiment to not be biased. Such factors where we keep the same, okay, we call such factors control controlled variables. We call those factors controlled what? Variables.
Very.
Or variables we keep what? The same.
Okay.
All right? Yeah.
Okay. Yes.
Now, there is another variable that we need to also take note of. There's this variable we call the independent variable. I don't know if you've heard of that before.
Independent variable.
Variable. Have you heard of that before?
No.
So in every experiment, there are three variables that usually decide how the experiment go. The first is independent variable. The second is dependent variable.
Okay.
And the third is controlled variable, or variable kept constant, or variable kept the same. Now, when we say independent variable, it means a variable, usually it's one most times, sometimes it could be two. The variable that you change as an experimenter, or in an experiment, the variable you change or you manipulate during your experiment. That's what we call the independent variable, the variable you alter. For example, in this experiment, what are we checking out? The effect of light intensity on the rate of what? On the rate of what?
Photosynthesis.
Yeah. So what variable did we manipulate or change or alter here? What did we change in this experiment?
We changed the position of the light.
The distance of the light, that's the light intensity. So that value, that factor that we alter, that we change, is the light intensity. So that becomes our independent variable. Now, the dependent variable is usually our result, what we measure as result, what we measure as outcome. That becomes the dependent variable. It's usually dependent. The dependent variable is usually dependent on the independent variable, because as the result keeps changing, the changes you are observing in the results is as a result of the change of the independent variable. So your result or your outcome, or what you measure during the experiment as outcome, becomes what we call the dependent variable. Do you get the idea?
Yes.
Now, the variable controlled or kept the same, of course, is the other factors that if you alter them, they can affect the reliability or the accuracy of your experiment. So we keep them the same. Those are the variables we keep. Are you with me?
Yes.
Any question?
No.
Can I proceed?
Yes.
Because I'm going to give you a question to answer now. That's how I'm doing this, all right? And I want you to answer it gallantly, all right?
Okay.
You with me?
Yes.
Good. All right. So let me see what we have next. Okay, let me just quickly explain this and we'll go to the question straight up. All right. Now, this experiment is also similar to the one we've had. Look at this setup now and tell me what you think is happening here. Just look at the setup, the apparatus setup, and tell me what you think is happening here.
Um. They have positioned the lamp a bit close um to the plant. So
Hmm?
this. Oh.
So what does that tell you? That with time, the plant will start photosynthesizing. Since light is what available, the water
Yes.
plant, of course, will have water and there's carbon dioxide in the water. So photosynthesis happens and number of bubbles can be counted, okay, or we can measure the volume of gas produced. So the more gas produced, the more space you find at the top part of this inverted test tube. So the more the gas produced, the more the gas will displace water and the level of water will keep moving downwards, moving downwards. So how can we measure, okay, or collect gas produced during an experiment? Usually a question they ask. We can easily count the number of bubbles given up per minute. A more accurate method is collecting the gas produced. And how do we collect the gas produced? We can collect it over water because gases are less dense than water. So they will displace water and collect at the top of the tube. So we can measure the point or the distance of the tube. Let's say this is 0 cm cube. This is 5 cm cube. This is 10 cm cube. We can measure the volume of gas produced. We can also collect the gas, okay, using a gas syringe. These are ways of taking or measuring volume of gases produced. Is either you measure the volume of gas produced in a measuring cylinder when you collect the gas over water, or you connect a gas syringe to the setup and you collect the volume of gas in the gas syringe, okay, where you can then measure the volume of gas produced from the syringe. So these are ways of measuring gas produced during an experiment. So they can ask you, in this experiment, the students counted the number of bubbles, but it's been inaccurate. What are the inaccuracies that can be associated with counting number of bubbles? What do you think could be inaccurate as to counting the
Um,
number of bubbles?
so a one bubble can merge together with another bigger bubble. So
Thank you.
if they don't count that one, their results might not.
Then the lunar is not accurate.
Yeah.
Beautiful. Sometimes the bubbles are not of same sizes. Too. Okay.
Mm.
Yes. And you can omit certain bubbles. You may not have accurately count all the bubbles given off. But if you collect the volume of gas, it's more accurate using a gas syringe, okay? Or you collect it in a measuring cylinder over water, then you can measure the actual volume of gas produced, okay, per unit time. That's why it's a more accurate method of determining the gas produced during an experiment. Do you get that now?
Yes.
So they could talk about how to collect the gas. We can collect it over an inverted test tube. We could also use what we call a delivery tube. Have you seen the diagram of a delivery tube before?
Yeah, that makes
Do you know what a tube looks like? Okay, let's quickly check through. Yeah, that's what it looks like. Okay.
okay.
So that's a gas syringe over there. Okay. So this tube connected to this setup is a delivery tube. It takes different shape. This could be a delivery tube. That could be a delivery tube. All right. So tubes they connected to a setup where there is a rubber cock to keep the gas from escaping. Then this tube collects the gas and you have the gas syringe measuring the volume of gas produced. So as the gas is being taken up, the syringe keeps pulling back and you can easily decide how much volume of gas there is in the tube. Do you understand? So that tube is a delivery tube for an experiment. Any question?
No.
So sometimes in some multiple or practical questions, they can actually give you a diagram and ask you to complete the diagram for the delivery tube setup, and what you just do is draw a delivery tube to it. So if you have okay, let me just show you what I mean so I don't assume you understand. Okay. So in a setup like this, for example, let's say this is the plant. That's the plant. Okay. And they'll just fix a delivery tube here. All right. And they'll connect a syringe to it. So that's the delivery tube. Okay. This is the gas syringe. Okay. And that's the setup, the plant. So the delivery tube is just to ensure that the gases or the gas produced is channeled into the syringe to be able to collect the actual volume of the gas. So such experiment can be asked you whether in paper 6 or in some. So it's just to have a wide knowledge of what is involved. Any questions so far?
Okay.
Any
No.
question?
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have um, you know the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, um, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable, and you get the very best of service. We also have for the E levels as well, um, and also for the EdExcel. So we have for the Cambridge E levels and EdExcel E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at agile@gmail.com, and they'll reply to you. And the good news about this is what I've discussed with the sales team is we've tried as much as possible to see how we can subsidize the amount according to countries, so it doesn't really look very, very heavy. Okay, so this is really important. Yes, we do charge in pounds, but again, we try to be as much as possible considerate to your countries, and we don't just skyrocket the price. No, because at the end of the day, we know how it feels, and we really want you to actually get the best of service, and again, be able to afford it from the comfort of your home. So again, Ajiro Tech private tuition is currently ongoing. You can enroll yourself for the IGCSE, and then for the E levels. Again, for the IGCSEs, these are the subjects, and for the E levels, these are also the subjects. So again, what are you waiting for? Just send an email to the system. If you're an IGCSE student preparing for IGCSE, if you're watching this in 2025, when this is in 2026, when this is 2027, we're currently ongoing, and this is actually a good time for you to actually explore, and then get it tailored to your own needs. So trust me, you don't want to miss out.
Okay, let's quickly answer a few questions. So at least we see how much you understand what we've done. So let's go. Which question should I do? Let's do some theory questions now. Look at this. Okay. So you're going to answer this question for me. Are you ready?
Yes.
Okay, since the students investigated the effect of different wavelengths of light on the rate of photosynthesis of the water plant Cambodia or Camboba, Abi
Sir,
Kabumba.
please, I just noticed something. Worksheets that you have, that's what I had for my test.
This particular one.
Yes, exactly.
Ah, interesting. Hope you were able to do well on
No,
it.
but
Yeah.
um the question that you're
Correction is
asking me now, I did kind of well in it.
mm, yeah. So do I look out for another question?
Um of So can you go down? Let me just see it. Okay, it's the same thing. So on that question B, I got 2 out of 3, but on that C, I didn't get anything. I didn't get any mark.
Okay, usually anytime they say calculating rate, rate has to do with distance over time,
Over
right?
time. Yes.
Yeah. So if you look at the or volume, anything whether distance, okay, or volume or whatever parameter is being considered in relation to time. Now here we are looking at volume of gas collected. So that's what you have over time. Look at what they said now. They said the student collected the gas produced by the plant for what was the time
5 minutes.
consideration now? 5 minutes.
5 minutes.
If they ask for the rate of photosynthesis, of course, it's the volume of gas collected per unit time. So each of these volume will be divided by what? Pi. So it's volume over time. And the time there is 5 minutes. So the volume divided by 5 minutes. Good.
Okay.
Yeah? Yeah.
Okay.
How about this one?
Oh, question D1 and DI and DII. I didn't get it right.
But now you should be able to get it right, right?
Yes. Why states why the students kept the lamb at the same distance during the investigation. So the light intensity won't be affected,
Mm-hmm.
or like, oh yeah.
Remember, they are looking at different wavelength of what light. Alright,
Mm,
so that's the light with color what filter. The color filter is what they use to change the wavelength of light. This color
mm,
filter can have different colors. When they put different filters of colors, it's going to change the wavelength of the light. But the light has to be at the same distance, so to keep the intensity the same, to ensure that the amount of light getting to the plant is the same. Okay,
okay.
so so it's the color filter that was being changed. All right.
Okay.
All right, so that's that with that. And they said, why did they use sodium hydrogen carbonate solution? I told you something about it.
Um.
What did I say sodium hydrogen carbonate does? And you add it to water, usually for water plants. What does it add to the water?
Carbon dioxide.
Thank you. It increases the amount of carbon dioxide available. So why do we need to add carbon dioxide? So that carbon dioxide is not a limiting one
Factor. Okay. Okay.
factor. Do you get now? So it
Yes,
could
it's all
affect the
coming in
rate of
place.
the yes. All
I
right.
got number
Okay.
2A correct.
Okay. Very good. Very good.
Oh,
Very
actually, sir, this is
good.
a different one. It's just the first from number 1.
Yeah, that
It's
one that
just
was
number
given
one.
to you. Okay.
Okay. Yes, it's different.
Okay. Let's try this one. Explain why the students included the following in the apparatus: the beaker of water and the thermometer.
Hmm. Um, the beaker of water to measure the amount of water that. is yeah.
Look at this. So there are two containers containing water.
Yes.
This is a water bath, and that's the beaker of water.
Yes.
It's used as a what? Water bath.
Yes.
Do you know the importance of water bath?
Oh yes. We did this in class. So I
Mm-hmm.
can't remember what solution that we put inside the test tube. It was basically to
Mm-hmm.
turn the plant blue black. So for um to hey seriously, what am I saying? You basically put the test tube inside a beaker of water, because if you heat it up directly, it can have like a reaction, it can like explode. So doing it with water is safer.
Okay. All right. I get that. That's you're referring to ethanol when you want to test the leaf for presence of starch. I get that. But this is somewhat different. In this experiment, you have light, okay, coming from the lamp, and there is a beaker of water, okay, and there's another test tube containing the plant and water containing diluted
Oh,
carbonate.
so the water and the diluted sodium hydrocarbonate solution will mix together.
That's one. But essentially here, this is to regulate the what? To regulate the what?
Oh, that's
Temperature
interesting.
of the heat, yes. The heat to
Oh,
that's what this is for. This water bath is to keep the temperature what? The same across the whole water region. Okay,
okay.
since the light is coming from one side, if it shines directly on the plant, it can affect the experiment. So this beaker is just to try to what, redistribute the heat to ensure that the heat is distributed and is the same across the plant. What's thermometer for?
To basically measure and check the temperature that the water. Oh
Temperature,
no,
yes,
that
to measure
the
the
dilute
of the
fluid
system.
is.
Yes, yeah. How about light meter and ruler?
Light meter and ruler. Um,
Common sense. Light and meter. What does that mean?
is it basically eh,
To measure the amount of what? Light
light?
that is emitted from the lamp. Simple. A meter measuring amount of light emitted from the lamp. How about the ruler?
Or
How
to
about the ruler?
calculate to measure the distance between the lamp and the
And the
station.
plant or
Oh,
the network. Yeah. Yeah. So that's it. So most times you ask questions like this a lot. Just
okay.
look at the experiment and figure out what apparatus, okay, what factor, what an apparatus actually, what significance each apparatus hold based on the idea painted by the experiment. Usually, anytime you have a question like this, the first thing that should catch your attention is the aim, the aim of the experiment, what the experiment is about, what the investigation is about. They said the student carry out an investigation. What is this finding out? The
To
effect
find
of
the effect
carbon
of
dioxide,
carbon dioxide concentration.
yeah, on the rate of photosynthesis. So we want to see how carbon dioxide affects. So most times the answer to supply will relate to how carbon dioxide affects the rate of photosynthesis. So always take note of this. With time, I'll prep more on questions like this. So I think uh okay, let's go back to our lesson so that we can answer more on limiting factors. Let me also say here, sorry, or am I being too fast?
No.
Okay. Okay. Let me also say here that you've seen that when you add sodium hydrogen carbonate to water in which there is a plant, you are trying to increase the what? Amount of carbon dioxide available. Okay? But there's this other compound called soda lime. Okay? When you add soda lime to an environment where you have a plant, soda lime actually absorbs all the carbon dioxide present around. Okay? Soda lime is the same as sodium hydroxide or potassium hydroxide. Okay? This compound absorb carbon dioxide. So if there's any carbon dioxide around any environment, immediately you bring soda lime close to it, it's going to absorb all the carbon dioxide around the environment. So looking at this setup, this plant is covered with a transparent bag, okay, and the air within it, of course, is exposed to soda lime. What do you think will happen within this bag? All the carbon dioxide around this plant will be what? Absorbed, isn't it?
Yes.
So if you expose this plant to light, probably after 3 hours, you are going to expect that photosynthesis will happen. But photosynthesis can't happen because all the carbon dioxide has been absorbed. And carbon dioxide is a raw material for photosynthesis. So after 3 hours, if you pluck one of these leaves and test it for presence of starch, it will not turn blue-black, showing that carbon dioxide is necessary for photosynthesis.
Yes.
So this is a simple experiment to show that carbon dioxide is needed for photosynthesis. Do you get the idea now?
Yes.
So there's a difference between soda lime and sodium hydrogen carbonate. This one adds CO2 to water. This one adds CO2 to water. But this one, which is soda lime, it removes CO2. Okay?
Okay.
It removes CO2. So don't mix them. You get the idea now.
Okay. Yes. Yes.
You get the idea.
Yes.
Finally, limiting factors. I've already explained what limiting factors are, and you've been able to give me different examples of limiting factors. So, limiting factors that are environmental, light intensity, carbon dioxide concentration, water availability, temperature. Yes, they all affect the rate of photosynthesis. And the ones that are internal, okay, could be amount of chlorophyll present in the leaf and also the surface area of the leaf. Do you get that now?
Yes.
Now, this is where they ask questions. Look at this graph. At the start, what do you notice it's happening as far as the rate of photosynthesis is concerned? As carbon dioxide concentration is increasing, what happens to the rate of photosynthesis?
Um. It doesn't increase. So it just
It's
stays.
not what increasing.
It's not.
As we increase carbon dioxide concentration this way, the rate is also what increasing. But at this point, what happens to the rate of photosynthesis?
It stops and
After this
just
point, it stops increasing and it stays what?
listen. Concentrate.
It stays
Yeah.
or constant. What do you
Constant.
think is happening at this point of X? At this point X, it means that any further increase in carbon dioxide no longer increases the rate of photosynthesis. So at point X and forward, it means that carbon dioxide is no longer what? It's not what?
Um, a limiting factor.
Is no longer a limiting factor. So from point zero all the way to point X, carbon dioxide is a limiting factor. But from point X all the way to point P, carbon dioxide is no longer a limiting factor. Something else is what? Limiting the rate of the experiment. Do you get the idea of limiting factors now?
Yes.
Sometimes they can have two factors at play at once. Look at the second idea or scenario. We are considering light intensity. We're also considering concentration of what? Carbon dioxide. Now look at this graph. There are two graphs, graph Y and graph X. Graph Y is the one with the red ink. Graph X is the one with the blue ink. Now look closely. Light intensity is constantly being what? Supplied. But something is being altered here, the concentration of carbon dioxide. So at Y, what is the concentration of carbon dioxide here at Y?
At why it increases, but then at a certain point it stays constant.
What is the concentration of carbon dioxide given to the plant here at Y? Look at it on the highlighted circle.
Oh,
How many percent carbon dioxide is supplied?
0.04%.
Percent. And at the blue mark, how many percent carbon dioxide do we have?
0.4.
What's the difference? Which one is higher?
The X is higher.
The X is higher. That's why if you look at the rate at Y, the rate became constant faster at Y because the concentration of carbon dioxide supplied was a limiting what?
Factor.
Okay, so at Y, what is limiting for this graph? Carbon dioxide concentration. So immediately, how can you tell from this graph? Because when the amount of carbon dioxide was further increased from 0.04 to 0.4, the rate what went up.
Yes.
Do you get it? So at X now, what can you say is the limiting factor? If it's not carbon dioxide, then something else could be what limiting. It could be temperature, it could be another factor. But at X now, we can say carbon dioxide is now what limiting. The only way we can be sure is if we increase this, if we do like if there's another graph now this way, and we now have this as 1% CO2. If this is 1% CO2, then what can you say is the reason for the graph staying constant at X? It means carbon dioxide is a limiting what? Factor.
Factor.
How can you tell from the graph that carbon dioxide is limiting? Because when you further increase the concentration of carbon dioxide to 1%, the graph further, the rate further what? Increase. Do you understand now?
Yeah.
So at X and Y, carbon dioxide are both limiting what?
Factor.
Factors. Do you understand the idea now? Since light
Yes.
intensity is constantly being supplied, so we can have two factors coming at play at the same time. So answer this question for me now before we go. Yeah?
Um, Carbon dioxide concentration.
Why?
Oh, why? Um, it's because as seen in the other line that when carbon dioxide increases,
Mm-hmm.
the rate of photosynthesis also increases. That means that the second
Very,
and the second one
very, yeah,
have carbon dioxide as a limiting factor.
very, very good, very good, very good. That means you get the gist properly. So that's how limiting factors can affect the rate of photosynthesis. Sometimes two things can be applied at the same time. Okay, look at this question. All right, look at this question. This is what I was trying to explain earlier as to how temperature affects the rate of photosynthesis. That temperature, when it's at the start, let's say this is 0 degrees C, this is 37 degrees, okay, and this is 60 degrees. Now, at the start, as you increase the temperature, the rate is increasing. Okay, this is in plants. So we use for plants, the optimum temperature for plants is around room temperature. So let's say this is 27 degrees C. And this is, let's say this is 35. Okay. So when you increase the temperature gradually from zero, the rate is increasing. And the rate is highest at the optimum temperature, which is 27. But just above the optimum temperature, what do you notice? The reaction starts dropping and the enzymes are getting what? The
Nature.
So at 35 grade, the reaction has fallen back to 0 degrees, meaning the whole enzymes, the active site are totally what? Denatured.
The
All right.
nature.
The rate of denaturation is faster. Okay. The difference in the temperature between the time for denaturation is narrower, is smaller than the difference in the temperature from the start to the optimum. That's how to differentiate between the graph of photosynthesis and pH. Do you understand what I've painted for you here now?
Yes.
Meaning above optimum temperature, the enzyme denatures very fast within a short range of temperature between 27 and 35. What's the difference? I think that should be around what's the difference between 27 and 35? That's about 8 degrees C, right?
Yes.
That's the difference there for denaturation after optimum. But from 0 to 27, that is what? 27 degrees C difference. Okay, so it means enzymes denature faster above optimum temperature than reaching optimum temperature. We get the idea. So that's how to differentiate for temperature and other factors and pH. Now answer this question for me and we'll call it a day. Look closely and be careful in choosing your answer.
A.
A, all right. He said what limits the rate of photosynthesis at point P? Of course, this is point P. So at point P now, look at this. The temperature here is 20. The percentage here is what? 0.1
Oh.
carbon dioxide.
Yes.
And here the
Okay.
temperature is 30. Percentage is 1. Here
Okay.
the temperature is
C.
20. Percentage is 2. Do
C.
you still want to stay with P?
No. C.
Uh, C, right? Because
Yes.
what shows that from the graph? Because light intensity is not limiting. There's enough light. That's what this is saying. What is being altered here is temperature and CO2. So
Mm,
here it means that at higher temperatures, the rate can increase. At
okay.
higher carbon dioxide concentration, the rate can also increase. So the two things that are limiting here are carbon dioxide concentration and temperature. So the
Okay.
correct answer there is what? C. Do you get the gist now?
Yes.
All right. So that will be it for now. Um, I'll see you tomorrow same time. Okay?
Okay.
Yeah. Have a good evening.
Okay. Thank you. Bye bye.